Role of mediators in transcription initiation in eukaryotes. For basal transcription, one does not really need a mediator, but to stimulate or enhance the transcription activity, or even the other way around, that is, decrease the transcription activity, play a very important role. So, in this session, the focus will be on the structure of the mediator and the way it is proposed that the mediators do influence transcription activity. Mediators are a protein complex that influence the transcription activity by acting as a molecular bridge. So they act as a molecular bridge between distal regulatory elements and the transcription machinery itself. And interestingly, these mediators are found ubiquitously right from the yeasts to humans. They are bridges, as mentioned, between the transcriptional activators or repressors, and they are basically influencing the RNA polymerase II to initiate transcription. Mediators are not considered as general transcription factors, as just mentioned. For basal transcription, one does not need the mediator. The general transcription factors are enough to actually uh, carry out or help the RNA polymerase II to initiate transcription. Although what has been found is that there are several genes that do require the mediators as they stimulate the GTFs to initiate transcription. Mediator complex is associated with the CTD tail of the RNA polymerase with a key regulatory function of the CTD kinase activity of TF2H. So suppose this is the pre-initiation complex that is associated with the core promoter region of the RNA polymerase II. The mediators basically help this entire transcription machinery to carry out or initiate transcription from moving from the pre-initiation complex to the initial transcription complex. So it's observed that you have the mediator coming and attaching to the pre-initiation complex at several points, with of course it having efficacy to bind to the CTD tail and also to the TF2D for that matter. And what is observed is that associated with the mediator binding to the pre-initiation complex is the looping of the DNA, because several transcriptional activators that are bound to either promoter proximal elements enable binding of or enable influencing transcription through the mediators. Similarly, you can have cohesin coming and forming the DNA loop or stabilizing the DNA loop to ensure that the mediator is able to influence the transcription activity. Moreover, you can also have the activators or repressors that are bound to the enhancer or the silencer elements. So these are basically DNA binding activators or repressors, and they on one side are binding to the DNA per se, where you have the cis acting elements. On the other end, they are binding to the mediator itself. So it is basically the cis-acting elements through the transcriptional activators that are able to bind to the mediators to influence transcriptional activity. So therefore, effectively, the mediators are forming a bridge between these transcriptional activators, repressors, and the pre-initiation complex. So they can be therefore considered as co-activators or co-repressors. And for certain genes, mediators seem to be a must. So therefore, let us look at the structure per se of the mediator complex and relate it slowly and gradually to its functioning. So it has been associated with the CTD tail of the RNA polymerase II. In fact, in yeast, the first initial four suppressor proteins that were found, SRB1 and 4, were found to influence transcription activity, and gradually from 4 subunits, it was found that the mediators actually are as large as 20 subunits and more. So from an early 4 subunit mediator, one has been able to find out that the mediator actually comprises more than 20 subunits, whether it is yeast or it is the humans. It helps in assembly of the pre-initiation complex, that is, it helps stabilizing the pre-initiation complex. It helps in what is called as the promoter clearance, so therefore basically helping in transcription initiation. And as mentioned, it is a bridge between the DNA binding transcription factors and the transcription machinery. So when you look at the structure of the mediator complex, it has been found that it can be subdivided into four main modules, and these four modules are the head module, the middle module, and the tail module, and with an outer CDK8 kinase module. So these are the four modules that are associated or that basically form the structure of the mediator complex. And the head, middle, and the tail modules form what is called as a core mediator, while the outer CDK8 kinase is a module that may be present, may not be present, or may be bound, may not be interacting with the core mediator. So when you look at the structure of the core mediator, you can see that every module is made up of seven different subunits, and these subunits are denoted as MED1, MED2, MED standing for mediator. So you have anywhere between 20 to 27 different subunits of mediator, ranging from across east to even humans. So, and these subunits that are there, they are basically localized into a certain module, and so their functioning will be only with respect to that particular module. You can see that you have the outer CDK8 subunit, which is made up of basically four subunits that can reversibly interact with the core mediated person. This is the transcription machinery to which you can have the mediator in binding. So, elucidating the structure of the mediators. So, the earliest structural component that was identified was the middle region of the mediator, and within the middle module of the mediator was found that it is made up of certain long helices with a flexible hinge in between, and interestingly, this flexible hinge is conserved right from species to species. So that's something that has been observed. So there are a lot of structural features that are conserved across different species. Next was identified or studied or elucidated what is called as the head module. And interestingly, the head module itself is subdivided into domains. So you can see, actually not domains, you can look at modules itself. So within a module, there are submodules present. So you have the neck region. So some of the MED subunits are able to form the neck region of the head module. Some of them are able to form what is called as a fixed jaw, and some of them are able to form what is called the movable jaw. And when they compared it to, the head module of different species, you can see that they were able to find a lot of similarity between the head modules that of mediators present in different species. This is the outer CDK8 module. So you can see that it is made up of four different subunits: the MED13, the MED12, the CYC C, and the CDK8. So the CDK kinase module per se is something that was discovered much later than the head and the middle region, but it was found that this CDK kinase module is able to interact reversibly with the core mediator region. So you can see that when the human MED, that is the human CDK8, the human core mediator was compared to the yeast mediator, one could find a lot of conserved structural modules in them. This is the association of RNA polymerase 2. So you can see that this is basically the primary, the initiation complex, that is a pre-initiation complex with the RNA polymerase 2, TBP, TF2B, TF2F, the TF2H, everything that is basically interacting with the mediator region. So this is a structural, um, uh, you know, uh, structural identification of how the mediator is able to interact with the pre-initiation complex. Now, basically, one when looks at the RNA polymerase 2, it has been found that several different parts of the modules of the core mediator is able to interact with RNA pol 2 and the pre-initiation complex per se. So we specifically TF2B has several contacts that it can make with the mediator. TF2D also can have several contacts that it can make with the mediator. The core mediator is therefore a region that is able to bind to the PIC, not the outer CDK8 kinase module. That does not really form a direct interaction with the pre-initiation complex. Now, the jaw of the head module of the core mediator is found to be interacting with the stalk, that is the RPB4, RPB7 of the RNA polymerase 2. And this interaction, interestingly, changes the conformation in the RNA pol 2 in such a way that the active cleft center opens up. Now, this basically opening of the active cleft center is actually looking at the fact that now the RNA pol 2 can accommodate the transcription bubble. So, both the strands within the active cleft center with the template strand basically bound in such a way that it is clear, it is near to the catalytic regions of the active cleft center. So this is a very important function of the mediator, that it is able to open up the RNA pol 2 active cleft center. Another interesting aspect with respect to the mediator is that the head module itself positions the RNA pol 2 CTD in proximity to the TF2H. So within the pre-initiation complex itself, the positioning of the pol 2 CTD to the TF2H is helped by the head module of the mediator. So therefore, that interaction also ensures that the kinase 3 subunit of the TF2H is able to phosphorylate the CTD tail of the RNA polymerase 2, which is important because that helps in further RNA processing as the synthesis begins. Now, this also is basically, with the CTD phosphorylation, one also observes that there is dissociation of the mediator from the CTD tail, leading to promoter clearance. So these are interesting interactions that have been observed. Another fact that has been observed with respect to the mediator structure is that not many of the subunits actually have enzymatic activity, but the outer CD8 kinase does have a kinase activity present. Now, suppose this is the core mediator region with the tail, middle, and the head portion. Then what is observed is that the outer CDK8 kinase can reversibly interact. So you can see that when needed, it can remain detached from the core mediator, but when needed, it can actually interact. And the CD8 module, okay, therefore can be reversibly bound to the core mediator. And with its binding, it brings in a dramatic change in the conformation and therefore the function. So the MED13, which is part of the CD8K module, is the mediator link of the core mediator with the CDK8 module. The CDK8 module is actually functioning in response to different signals in different cell types. So it is able to enable the mediator to act tissue specific way. The different, and in fact in the same manner, because you know that every module is made up of several different mediator subunits, it is interesting to note that almost all the subunits present in these modules of the core mediator are able to bind to different signal molecules or respond to different signal molecules or bind to different transcription factors, thereby enabling the mediator to influence the initiation process in response to these different interactions. So the conformational changes that happen because of the different subunits binding to different, activators, repressors, or signal molecules it would either affect the transcription in a way that the transcription is decreased or the transcription is enhanced. What has also been noted is that post-translational modifications of the subunits present in the core mediator can also play a role in its functioning. Now we will just look at as a flow of how the mediator is basically trying to act as a co-activator or a co-repressor. So as you can see over here, when the transcription activators or repressor are either bound to the enhancers or the repressors, which are cis acting elements that are found distally from the promoter region. So you can see how the core mediator region is actually bound to by the transcription activators, and at this precise time when it is bound to the transcription activators, you can see how the DNA has to loop in order for the mediator to actually bind to the transcription activators. Plus, you can see that when the core mediator is not yet associated with the transcription machinery, the outer CDK8 kinase module is associated with the core mediator. Now, when you have the pre-initiation complex formed in the promoter region, you will see that the outer CDK8 kinase module is detaching itself from the core mediator, and it is the middle region of the core mediator that is able to interact with different parts of the pre-initiation complex. Like, for example, it is able to interact with the TF2D. You can see that the tail region is able to interact with the TF2B. You can see that it can interact with cohesin, and cohesin is something that is stabilizing the looping of the DNA. You can also observe that the core mediator is ensuring that the CTD tail of the RNA polymerase lies close to the TF2H, so that the TF2H is able to phosphorylate the CTD tail, because the TF2H has a domain which has CDK7, or what is called as the kinase 28 kinase of the TF2H, and that is able to phosphorylate different amino acid residues present in the Pol2 CTD. So these are things that happen as you have the mediator bridging the pre-initiation complex with the transcription activators. Now, after this, what happens is that once you have the RNA polymerase initiating transcription and moving away, so moving away from the promoter region, you can see very well that there is what is observed is a clear promoter clearance, and with the promoter clearance, therefore what happens is you have the mediator region dissociating from the RNA polymerase, and the moment it dissociates, you can again have the outer potential CDK8 kinase module reassociating with the mediator. So in a way, the outer module is kind of responsible for whether the mediator is bound to the pre-initiation complex or not. So when it is detached, it can bind. When it is not detached, it is not bound. So that confirmation that the mediator takes when the outer CDK8 kinase module binds is different from when it is not bound, and that makes the difference. So although a lot has been found out about the mediators, there is a long way to go to find out that about every subunit that is associated with the mediator. And interestingly, what has been observed is that when the mediator is deficient of one or two subunits, it has affected the functioning of the mediator, and more so many of the mediators, when they are either defective, that means mediator subunits, when they are either defective or there is loss of function, then it has been associated with several human diseases, especially cancer. So therefore you can understand how important the role of mediator is in the transcription process of the eukaryotes. So with that, let us make the conclusions. Mediator, comprising more than 20 subunits, has these subunits arranged into head, middle, and tail modules, with a fourth CDK8 module present as a reversible detachable module. The core mediator forms a bridge between the transcription activators, bound to the distal regulatory elements and the pre-initiation complex at the promoter region by looping of the chromatin. The core mediator is able to bind to the stalk of the RNA polymerase II to open the active cleft center, which takes forward the transcription initiation. The mediator positions the CTD tail of the RNA polymerase II such that the TF2H kinase activity is able to phosphorylate CTD tail through its CDK7 kinase 3 domain. So mediator subunits are able to regulate transcription as its individual subunits are able to bind to different molecules that in turn are able to interact with the pre-initiation complex to enhance or repress transcription. Loss of subunits have been associated with several diseases. Thank you.